Welcome to the Rhythm of Life. We're going to celebrate DNA Day today. Center for Excellence in Education is a Virginia-based nonprofit founded in 1983 by Joanne DeGeneiro and Admiral Rickover, who's father of the nuclear Navy. And our center has a mission to nurture high school and university students to careers of excellence and leadership in STEM. Our center's programs, we have the Research Science Institute, the USABO, Teacher Enrichment Program, which is hosting this event tonight, as well as our new program, STEM Lyceums. I am pleased to welcome or introduce our two speakers who will be helping us celebrate DNA Day. We have Dr. Christopher Hopkins, and we have Dr. Michelle Gumz. I'm going to read a little bit about them for you. Dr. Christopher Hopkins is an executive sequencing assistant or specialist, at Illumina. His background is in molecular biology and assay development, genomics, microarrays, and DNA sequencing. Dr. Hopkins has held research, assay development, and commercial positions at several technology-based companies. We also have Dr. Michelle Gumz, who is an associate professor at the University of Florida, where she also serves as an associate director for the Center for Integrative Cardiovascular and Metabolic Disease. She earned her Ph.D. in Biochemistry and Molecular Biology from the University of Florida and completed her postdoc training in renal physiology. Dr. Gumz and her lab study the role of molecular circadian clock in the kidney and its contribution to blood pressure regulation and renal function. So yes, I am pleased once again to welcome Dr. Christopher Hopkins and Dr. Michelle Gumz. First presenting will be Dr. Christopher Hopkins. He tells us a little bit about what he does at Illumina and DNA. So go ahead, Dr. Hopkins, I'm turning it over to you. We're excited to hear your talk today.
Thank you, Laura Ashley. Thank you for the opportunity. And I'm really pleased to be able to talk to you all today about something very near and dear to my heart, which is DNA sequencing and the genomics revolution that is being driven by DNA sequencing on this very auspicious day, DNA Day, April 25th. I want to start by just let's say, what is DNA Day and why do we celebrate DNA Day on April 25th? And there's two big events that occurred on April 25th. First was in 1953, was the published elucidation of the DNA structure, the double helix. And showing here the original publication in 1953 by James Watson and Francis Crick. And all of these folks worked at the University in Cambridge. But also Rosalind Franklin is actually not credited in part of the publication, but her data was really critical. She was an X-ray crystallographer, and this X-ray diffraction pattern here was very critical data for understanding the double structure. So I really want to call out Rosalind Franklin and her contribution to understanding the DNA structure, which was really not that long ago when you think about it, 1953. Fast forward to 2003, and that was the day on April 25th of 2003 that a draft of the human genome was published. So the human genome is enormous. It is 3.1 billion bases of sequence. And this was a monumental achievement and really just a really exciting time. That really just the beginnings of what we'd call genomics was the sequencing of that first genome. So I want to start with kind of a little bit of a personal note, which is how did I get into genomics and why do I think DNA is so exciting? And for me, it started with this, what we're looking at. So if you don't know what this is an agarose gel. And what you're seeing is these fluorescent little lines are fragments of DNA that have been separated by size, stained with a dye that fluoresces. And what we're looking at is little pieces of DNA of different sizes. And when I was an undergraduate at Humboldt State University in Northern California, where I live, I saw this and I was just blown away. I was like, wow, people are looking at DNA. And I saw this in a lab. I was so excited and I kind of knew what DNA was, but I had no idea that you could do things like cut it up and see it and manipulate it and analyze it And this really was a life-changing event for me. I was a biology major. I actually changed my major to biochemistry and molecular biology. I transferred to another university, University of California, Santa Barbara, so I could specifically study molecular biology and DNA. That's what I wanted to do. And that's pretty much the path I've ridden. And boy, we have come a long way. So let's start by talking about just some basic biology, which probably many of you don't already, if you're a biologist, you know this. But this is really key to understanding what is DNA and what is the biology behind it. So if you kind of start at the cellular level, our bodies and most organisms are made up of cells, right? And every cell has within it a nucleus that has the same copy of DNA in it. So every cell in your body has the same copies of DNA. And the DNA is composed of chromosomes, which are discrete units or particles, so to speak, of DNA. And if you unwind those chromosomes, what you end up with is this long linear strand of DNA. It's so incredibly long. It's so tightly wound and packed with these histone proteins into your cell. In fact, if you were to stretch all of the DNA from one cell of your body, which you cannot even see, if you were to stretch it end to end, it would be 3 feet long. So there's really a lot of DNA. And what each of these strands is composed of these 4 different building blocks: adenine, guanine, cytosine, and thymine. And so here they're shown as color-coded blue, yellow, green, and red. And guanine pairs with cytosine and adenine pairs with thymidine. So understanding this, the DNA sequence is actually understanding that how these different bases, they're linked together in a chain like this. What is the order of these bases? Because this order of the bases is really, it's the code of biology. It's the blueprint of everything of your entire life. You start off as a single cell, and within all of this DNA is essentially the information needed to make and grow and maintain and reproduce an entire organism. I mean, that alone to me is something of a miracle. But what is this DNA? What's in there, and how do we figure out what it is? Well, one of the first things you need to do is to decode the DNA. So if we discovered the structure in 1953, it wasn't until 1977, in the late 70s, that really methods to actually decode DNA became available. And a big, important technique that came out was developed in Fred Sanger's lab, again at the University in Cambridge, called dideoxy chain termination chemistry. And the principle of how this sequencing works is really pretty similar to what we do today, at least where I work at Illumina, the way our platform works there. And what you do is you start off with a little fragment of DNA that's shown here in purple. Hopefully you guys can see my pointer. And you use this as a template, and you essentially synthesize a complementary strand. And what you do is you end up with, you use these dideoxy terminators that essentially give you strands of varying lengths, and you have 4 different colored nucleotides. So you can separate them based on size and by fluorescence. So essentially you're decoding by synthesizing a complementary strand. This was an incredibly powerful technique. And when I was a graduate student, I sequenced a gene. It was 3,000 bases. It took me about 6 months. Today, you could do that in probably less than an hour. So Illumina did something really revolutionary in sequencing. And what they did is they figured out how to take this simple single reaction and really do it in a massively parallel way. And I just want to show you how the basics of how it works. You start off, you need a little fragment of DNA. So if you're looking at something really big, like a human genome, you break it up into a bunch of little fragments, right? And then we're going to sequence each one of those fragments, but we're just going to look here at a single fragment, okay? And what you do, you don't need a lot of DNA to do this, less than a gram, is you attach these little adapter sequences to the end of it. So there are little pieces of synthetic DNA. And there are 2 different ones. There's a red one and there's a blue one. And then we basically attach those by hybridization, because the double helix comes together. We attach these little fragments to the surface of what we call a flow cell. It's basically a piece of glass. And on that piece of glass are the corresponding red and blue attachment sites. So you attach them You then attach the other ends by flipping them over. You synthesize another strand. You do this enough times to generate what we call a cluster. So each of these that's one fragment here represented in each one of these clusters. And that fragment might be a few hundred bases long. And then what we do is we sequence those. And we sequence those by pretty much the same thing I showed you with the Sanger, where we have different dye-labeled nucleotides. We essentially take some polymerase, an enzyme, we incorporate a nucleotide. We then shine a laser and scan the little cluster, and then we measure the fluorescence and we say which dye was incorporated there, because there's 4 different dyes. So if you look down here in the lower left here, you see a little picture of a sequencing reaction. Each one of those dots represents a different cluster that's being sequenced, or a different fragment, right? So you reverse that, you add another dot, another base, you incorporate another base, and just repeat this over multiple times. And then you scan with a laser each time, right? So we're not just doing 2 reactions though, like I'm showing you here. We're doing billions of these in parallel, right? So think about that. I did my reactions in graduate school. I would do a single reaction, maybe get a couple hundred bases. Here we're sequencing billions of reactions at the same time, and then decoding them with a computer, basically. So this has really been a revolution in sequencing. Oh, I forgot to show you. That's how it's decoded. You look at each of the bases for each of the additions. So how has Illumina really played a big part in this? It's really these instruments that they make that basically do what I just showed you. So in 2006, they came out with a genome analyzer, and it was able to sequence a gigabase, or 1 billion bases of DNA, which was remarkable at the time. In 2010, they came out with the HiSeq. They sequenced 600 gigabases, or 600 billion, the HiSeq X in 2014. Basically, every few years, they've come out with a new instrument, and we're now able to basically sequence 3 trillion bases of DNA in a single run, which takes a couple of days, right? So if you look at how that's impacted the cost and the speed with which we can decode DNA, it's just amazing. So this graph here is very common. You'll see in a lot of next-gen sequencing or next-generation sequencing talks, where they show how the cost of sequencing a human genome has decreased over time, right? So when the first human genome was sequenced in 2000, started sequencing in 2001, it cost about over $100 million. That's the sequence of one genome. It took teams of people, lots of different instruments. It was a massive project. But Illumina came out with these instruments that increased output and decreased the cost over time. And these steep drops you see show that decrease in costs, right? Right now, you can sequence a human genome on the NovaSeq instrument for about $600 in consumables, and it'll take you about 2 days. So we've really come a really long way, and we've gone from being excited about sequencing a single human genome in 2003 to now sequencing millions of genomes each year. So that's cool. What do we sequence? What are we doing, right? If you think of a sequencer as just a giant machine, you put your sample into it and you get this, you can sequence a lot of things, right? Well, what can you do with that? This is the amazing part. And this is actually what my job is. I talk to people every day who say like, hey, we want to use your sequencer. We're trying to do this. How do we do it? And so that's really the interesting, fascinating part of my job is I get to talk to people doing all sorts of different things. So I'm just going to put them into these broad categories. Then I want to show you something specific that I think we can all really relate to. Genetic disease. Do you know that there are almost 1 in 10 people suffer from some kind of genetic disease, a mutation in one of their genes that knocks it out or affects it. And there are so many different genetic diseases. There's over 7,000 of them that we really don't know much about all these diseases. For years, people couldn't really even know what the disease or what gene was affected. Now we're able to go in there, identify people's genetic diseases, get them treatment, or start doing the research to help them find cures. Cancer. we all get touched by cancer, and we know of people, or we ourselves have been touched by cancer. Cancer is essentially a disease of DNA, mutations in DNA. We now can sequence genes that we know cause cancer or predispose people to have cancer, and not just find it early. We can match people with therapeutics and learn more about how cancer works. RNA expression. So you can sequence RNA. RNA comes from DNA. It's a gene sort of generates RNA. And we've learned so much about how genes are regulated and expressed through DNA sequencing or RNA sequencing. Understanding how proteins bind DNA. Understanding agriculture, how we breed plants. I mean, it's all genetics, right? So this is all assisted by sequencing and understanding. Microbiology and infectious disease. Microbes like viruses and bacteria, they have pretty small genomes compared to humans, and they're very easy to sequence. And I think really the best example I can share with you guys today of the power and the importance of genomics and DNA sequencing is something we all have been affected by. I wish I was in a crowded room and I could ask you all, who knows what this is? I don't expect you to know, but I'll tell you what this is. This is the sequence of the coronavirus. So in January of 2020, actually in December of 2019, Professor Yang Zhenzang at the Shanghai Public Health Center sequenced some samples that he got from an unusual outbreak in Wuhan. And they said, this looks like a coronavirus. So there was really, from the time that they identified the outbreak to finding the coronavirus was a matter of days, maybe weeks. Just for comparison, before this type of sequencing was available, the HIV virus took 3 years to identify. We found the coronavirus in a few days. And you know what that led to? It led to tests, the development of PCR tests to detect it. That came from the sequence. You couldn't do that without the sequence. The incredible vaccines, the RNA-based vaccines from Moderna and Pfizer, those came from this sequence, this very publication right here. In fact, the people at Moderna, they said, we've never even had a virus at our site. We've never even seen or touched a virus. Everything they did was from the sequence itself. So the discovery to the detection, to the vaccines, all of it centered around the actual sequence and determining that sequence. So it's really had a huge impact, and it's been really meaningful for me to work with public health labs who are sequencing this virus to find variants. All the variants are determined by sequencing. So that's been something that's been really exciting and fun for me that I've worked on a lot. So I just kind of wrap that up, and I know there's a lot of students here and you're thinking about what you want to do in the future, and I just want to put out Illumina there and just say that I've been there 9.5 years. It's been an incredibly exciting place. I've watched this revolution of genomics just grow every day. When I got there, it was about 2,000 people. We're now closer to 10,000. And it's a great place to work. If you're interested, I'm happy to talk to you. Put up a little QR code for those people who know how to get their phones out and want to see about our great internship program that we have in San Diego. I think we may have it at other sites as well. Anyways, I encourage you to seek it out. It's a really wonderful company, and encourage you to just enjoy the excitement I have around DNA. And with that, I'll just say thank you. And I'm going to put up my email address here if you want to reach out to me, and my Twitter handle if you want to join the conversation online. So thanks a lot. And I think we're gonna go to the next speaker. I'm really excited to hear about this circadian rhythms, and then we'll take questions at the end.
Absolutely. Thank you so much, Dr. Hopkins. That was great. We discussed earlier, I'm not a life science person, but this presentation was great. And I understand. Did I convert you at all? Yeah, no, I mean, a little bit. I appreciate it, and I understand it a lot better, and a lot, you are exciting. You would be a great, if you were my teacher, maybe I would have gone into life science instead of engineering, but thank you. All right, now we're going to turn it over to Dr. Michelle Gumz. She is going to talk to us more about DNA. Take it away, Dr. Gumz.
Hi, everyone. Good evening. I'm really excited to join this group today. Happy DNA Day, everyone. So I'm going to talk today about circadian rhythms and the molecular mechanism that drives our circadian rhythms. And then I'm also going to tell you a little bit about the kidney, which is my favorite organ, and show you just a little bit of data from my laboratory at the University of Florida. So circadian rhythms, the actual term circadian or circadian is Latin for about a day. And it is a cycle that repeats every 24 hours, not by coincidence, as long as it takes for the Earth to go through one full turn. And circadian rhythms are thought to help our body save energy through anticipating what's about to happen. So for instance, if we wake up and eat breakfast at the same time every morning, there will be enzymes in your digestive system that are waking up and ready to go right when you're ready to eat. So I do like to give just a little bit of a historical perspective because we've known about circadian rhythms for quite some time, actually. So if we go all the way back to 400 BC, this scribe for Alexander the Great noted that he observed the leaves of certain trees that were open during the day and closed at night. And then many years later, a French astronomer named Demarian observed that the daily rhythmic motion of the heliotrope, this plant that he had in his home, that those motions persisted even when he took that plant out of his windowsill and put it in a closet. So that's how strong these circadian rhythms are. And then if we jump up to the 20th century, back in the 1950s, they found that these starling birds use the sun to migrate, that their actual internal clock orients them as the sun changes. And then jumping up to 1994, this was when the first mammalian circadian rhythm gene was cloned, and it was called clock. And that work was done in mice. And then if we come up to 2001, this was when the first human circadian rhythm gene was discovered, with the identification of a family out in San Francisco that had this sleep phase disorder. And so they studied the members of this family, and they had this sleep phase disorder, and they all had in common a mutation in one of these circadian rhythm genes. This one was called PIR2. So circadian rhythms regulate a lot of our daily activities. It controls when we wake up and also when we go to sleep. It helps regulate our eating patterns. It also controls our cardiovascular efficiency. So studies have shown that for most humans, our highest reaction time and our best coordination and strength are in the afternoon hours. And so it turns out many organisms have circadian rhythms. This process helps animals know when to wake up, when to hunt, when to look for a mate, for example. It also helps them know when to migrate, like we talked about with the starling birds back in the 1950s. And it's not just animals and birds. We talked about Demarian's observation with the plants. It also helps plants know when to bloom and spread pollen. So going even now into simpler life forms, even bread mold and bacteria have circadian rhythms. So this is really interesting to think about. You may know that we have a microbiome in our digestive system where we have a balance of healthy bacteria, and those bacteria themselves have a circadian rhythm. So where do these circadian rhythms come from? So now I want to take us down to the molecular level, and we're going to look in an example cell. And so as our last great speaker described DNA and the discovery of its structure, I have here a little simple double helix. So this is just one example gene of many. This DNA is located in the nucleus. And these circadian rhythms are driven by a group of core proteins that regulate gene expression. And so one of these proteins is called clock, and I just told you about how it was discovered in mice back in 1994. It has a partner called BMAL1, and these two proteins interact with each other, and they bind directly to the DNA sequence in a sequence-specific manner. So this E box refers to a series of bases in the DNA sequence that these proteins recognize and interact with. And what they do here is they drive expression. So this is my little transcription arrow to make messenger RNA for their target genes. And two of those genes include the period and cryptochrome genes. These are additional circadian clock proteins that, when translated out here in the cytoplasm, then translocate back into the nucleus, and they actually interfere with the action of BMAL1 and clot. So this mechanism, where the one set of proteins binds DNA, activates the expression of the other proteins, Pierre and Cry feed back on and inhibit this action, this whole cycle takes about 24 hours. And this drives the expression of about 50% of all of our expressed genes in the whole body. And this clock mechanism is present in just about every single cell type in our bodies. So how did we learn all of this? Well, one of the most exciting events in the field of circadian biology was in 2017, when the Nobel Prize in Physiology or Medicine was awarded to Jeffrey Hall, Michael Rosbash, and Michael Young. And this award was given in 2017, but it was actually for work that they had done in the 1970s and the 1980s using a very basic research model, the fruit fly, Drosophila. And so they studied these flies, and they found flies that had mutations in their circadian rhythm. And they used that to separate the flies out into those that had a longer than 24-hour rhythm and those that had a 24-hour rhythm. And in doing that, they were able to use cloning methods to identify the gene that was driving this circadian rhythm. And it turns out that that gene is one of the period genes, PIR1, which is the focus of my laboratory. So what was also very exciting when the Nobel Prize was awarded in 2017 to those circadian researchers is that the Nobel Prize account on Twitter had a flurry of activity related to circadian rhythms. And so in this particular picture that they tweeted, this is depicting a 24-hour clock. So a little different than the clocks that we look at, on the kitchen counter, for example. Here, noon is at the top and midnight is at the bottom. And so what this is showing us are the different physiological functions that peak at different times of the day. So we have this hormone called cortisol, which is a stress hormone, and it has a peak early in the morning. The fastest increase in our blood pressure, this morning surge in blood pressure, occurs early in the morning. As I already mentioned, our reaction times and coordination are at their peak late in the afternoon. Blood pressure also peaks late into the afternoon and into the evening. Melatonin, which you may have heard of, some people use this to help regulate their sleep patterns. This is a hormone that's only secreted in the dark, and it is also related to the circadian cycle. So coming back to why we have these circadian rhythms, well, it's thought that this molecular mechanism evolved long ago. It's present even in bacteria all the way up to humans, because it provided organisms with an advantage living on a planet with 24-hour periods of light and darkness. And so again, this comes back to the idea that circadian rhythms help us predict what's going to happen and helps our bodies adapt in the best way possible. So now I want to talk about some tissue clocks. So I've already told you that that molecular mechanism for the circadian rhythms is present in all of our cells. And this is true across our organs as well. And so there is a central clock located in the brain, and it is directly regulated by light signals that travel through our retina directly to the brain. Food signals also tell our brain, as well as our other tissue clocks, and these include but are not limited to the kidney, which is my favorite tissue, the liver, and the digestive system. And so all of these clocks in a healthy individual are synchronized, and they help coordinate our rhythms in physical activity, sleep, wake, and also cardiovascular function. So blood pressure, which is one of the interests of my laboratory, has a very strong circadian rhythm to it. And just as an example of how important these circadian rhythms are to our overall health, there have been a number of clinical studies done over the years that show if you lose that circadian rhythm of blood pressure, it's actually associated with an increased risk to have a heart attack or a stroke. And so we know from large studies like that, that it's important to maintain a healthy circadian rhythm. And so when you hear people say you shouldn't look at your cell phone at night, this is why, because that light will travel through your eye and tell your brain it's time to be awake, when in fact it's time to be asleep. And so we can better manage our light exposure to help improve our circadian rhythms. So as I've mentioned, the kidney is my favorite tissue. So I want to take just a moment to talk about what our kidneys do for us. So the kidneys filter the blood, and they get rid of wastes like toxins, breakdown products from medication we might take. But we also get rid of other substances that we may not need. Like if you have too much salt in your diet and your kidneys are working properly, you'll excrete a lot of that salt because the body doesn't need it. Blood pressure is regulated by the kidney very carefully to make sure that it isn't too high or too low. Through its filtration and its excretion of substances like salt or sodium and chloride, the kidney also helps regulate body water. It helps regulate bone health as well because the kidney makes an active form of vitamin D, which helps our body absorb calcium. The kidney is also important for acid-base balance, so this keeps our pH levels in our blood at just the right level. And then the kidney also makes this erythropoietin, which tells our bone marrow to make red blood cells. And so you can see the kidney has a number of different functions, but what we focus on in my laboratory is how the kidney handles sodium. And so we measure in our animal models how much sodium is excreted in the urine. And we also look at what happens to blood pressure. So to do those types of studies, we use mouse models, and it turns out that mice look very much like humans in terms of their blood pressure, circadian rhythms, and their cardiovascular and renal function. So we use them to model what happens when, let's say, you delete or knock out a certain circadian clock protein, and we look at what happens to blood pressure and kidney function. So here's an example for the little bit of data that I'm going to show you. Let's say we have a control mouse. This is what we call a wild type, meaning all of its genes are intact. And this is what we're going to compare our data to. So these are our controls. Then we have knockout mice. And so I'll abbreviate those as KO or knockout. And in the data that I'm going to show you, we have knocked out the circadian gene PIR1. And we say global knockout because PIR1 is missing from every cell in the mouse's body. So we're going to see what that does to blood pressure and kidney function. Now, based on some earlier studies that we had done over a decade ago, our hypothesis was that this circadian clock protein, PIR1, regulates blood pressure and also sodium balance through the kidney. And the methods, we measure blood pressure in the mice, and we do that by implanting a tiny probe inside the mice, and they wake up and recover from that implantation surgery, and that allows us to measure their blood pressure in real time so that we can see those circadian rhythms, and I'll show you those in just a moment. And we also collect urine samples from these mice so we can look at renal function or kidney function by measuring how much sodium is excreted in the urine. So in this graph, I'm showing you blood pressure on the y-axis, and that's in millimeters of mercury. So this is for the control mice in orange. Each data point here is a 1 hour recording of blood pressure. So this would be as if for humans, if you had a blood pressure cuff on your arm and you walked around for about 48 to 72 hours, and we took readings every hour and we plotted that data. And if you did that for a healthy human, the data would look very much like what you're looking at right now for these control mice. The only difference is that mice are nocturnal. So they're awake during the nighttime when we're asleep. And so their blood pressure is higher when the lights are off and lower when the lights are on. And so if you see this pattern, it repeats over these couple of days. The blood pressure is lower during lights on and higher during lights off, because that's when these mice are active and awake. So that's the control mice. What happens when we take away one of the circadian clock genes? You can see some big differences in the blood pressure. And what I like to point out here are these big gaps during the time when the lights are on. And this is when the mice should be resting or sleeping. Their blood pressure is lower. And this is, again, regulated by circadian rhythms. To maintain a healthy circadian rhythm, our body needs our blood pressure to be lower during the night, for humans, the night while we're resting, and for mice during the daytime. And the other thing you can notice here, if you look at the blue line for the knockout mice, is that they don't have that decrease or that dip in their blood pressure when they should. And so this is associated in humans with bad cardiovascular outcomes. And so this mouse model gives us a chance to study whether or not we can fix this, and that might someday help humans who suffer from circadian rhythm disruption in their blood pressure. The other thing that we do in my laboratory is collect a lot of mouse urine. This is something I always tell potential students right up front. It is, a dirty job, but we take our kidney physiology seriously, and we learn a lot from these studies. So in order to do this, we have the mice in specialized homes where we can collect their urine. And what we do is we go in twice a day. So mice are small. They only have about 1 milliliter to 2 milliliters of urine in a day. So we get a little bit during the lights on when they're resting, and we get a little bit more at night when they're active. So this allows us to measure their food intake, their body weight, and collect their urine so that we can compare differences between the night and the day. So as I mentioned, it is a lot of urine. Typically, we have at least 12 mice in one of our studies. We collect 2 times a day for many days. And so you can imagine we generate quite a few tubes of mouse urine. And so I always like to give a shout out to our students in the laboratory, because this is very hard work. But we get some really exciting information from this, and we hope that it will help improve human health. So what does the night-day sodium excretion data look like? So in this graph on the y-axis is how much sodium is in those urine samples. And you can see going from the night to the day, how that pattern goes up and down and up and down. Again, this is very similar to what you would see in humans, except the pattern would be reversed. And so you can see some differences here, that in the Pure 1 knockout mice, they start to have some differences in their night-day pattern. So we calculated the ratio. So how much at night compared to how much they excreted during the daytime. And in the control mice, this was about a 7-fold difference, which is a good, healthy difference. But in the knockout mice, this ratio was reduced. And so what happened was that they were excreting more sodium during the daytime and then a little bit less at nighttime. And again, we think this is associated with a circadian disruption in the function of the kidney. So overall, these data have increased our understanding of how these circadian clock genes contribute to the regulation of kidney function and also blood pressure. And we hope that this new information will someday lead to new treatments to treat kidney disease as well as high blood pressure. And so I do want to give a shout out to the students and the postdocs in my laboratory. We have a number of just really amazing people. I'm so fortunate to work with all of them. This was during COVID. We gathered outside and had a celebration for some of our students who were graduating. So thank you so much for your attention, and I look forward to our discussion.
Thank you so much, Dr. Gumz. That was interesting. That's very interesting. And yes, your poor lab assistants who have to deal with all the mice urine. I can't imagine. Okay, so we at this time are going to do Q&A. So at the bottom of your screens, if you're attending and you have a question that you would like to ask either of our panelists, Dr. Hopkins or Dr. Gumz, please, you can type your question, submit your question in the Q&A, and we can discuss. But I guess I can start off by asking each of you, what is your favorite thing about what you do? What do you enjoy the most about your work?
I'll go first if that's okay. I think the thing that I love about it the most is that it's very relatable. So I'm, a basic science researcher. So a lot of times when I'm talking to people about what I do, it can get too technical. But when I say that we work on circadian rhythms, and I'll often ask people, are you a night owl or are you an early bird? And everyone can kind of relate to that. And all of that is tied to our circadian rhythms. So I think the relevance of it to human health and just, the general population is one of my favorite parts.
Yeah, I think my answer would be similar. I feel like what I'm doing makes a difference and is important. And, I gave the example of the coronavirus stuff because it's just so meaningful to me. And I felt my work was important. And the mission of Illumina is to improve human health by unlocking the power of the genome. I love that mission statement because I just believe in it so deeply. And that's really meaningful to me. What else do I also like about my job? I love working with the people I work with, just really, fun, smart people solving problems, and I get to, talk to scientists like Dr. Gumz and hear about their work. And I just love those interactions and helping people solve problems, and I just love tackling and figuring things out. So, yeah.
Thank you. And I had a question, and apologize if someone is going to ask this later, but I was thinking during your presentation, Dr. Gumz, about all because I just downloaded one myself, like a blue light blocker, like an app that blocks blue light. Does your lab do any work that sort of further explores like how that, when we have those blue light blocker apps, does that decrease? Does that help? Does it assist? And kind of like, are we able to measure that? So I don't know, have you all done any work exploring that?
Yeah, that's a great question. We haven't looked directly at that with the models that we use, but there is plenty of data out there to support that. I saw one study presented at a biological rhythms meeting a couple of years ago, where they tested the blue blockers on graveyard shift nurses at a hospital. And they found some improvement that it helped them, kind of get ready to go to sleep in the morning, in the daytime, which is incredibly difficult because humans are not meant to be, nocturnal. So there's definitely some data out there to support that.
Thank you.
Can I ask a little question too?
Absolutely.
First of all, I loved your talk, Dr. Gumz. It's really fascinating. And it's so relatable because everyone experiences this. And I was thinking about jet lag and how I'm affected by that. And I'm wondering, what's known about that. And at one point, will we have a drug that I can take that when I fly to Europe and I wake up, that I'll adjust quicker? Is there a way? What do you know about that?
Yeah, that is, it's something else that's so relatable. And I love talking to people about it. It's so fascinating. You'll have some people who say they get worse jet lag if they're traveling west versus traveling east. And it really, like the feeling that we get, whether it's a headache or even a stomach ache, is related to the fact that all of our body clocks become out of sync with each other. So you might land in Europe and the sun is out and your brain says, okay, it's this time. But your stomach and your kidneys and the rest of your internal body clocks are still on, East Coast time from the United States. So they're that is related to some of the symptoms that people feel with jet lag. And in terms of the basic research that's been done, we have used a model of jet lag with our animal models where we shift the light cycle to mimic what would happen, if you were traveling to Europe, for example. And we do see that that's associated with disrupting rhythms in kidney function. And so that's true for mice and for rats. And when you think about human studies, there have been quite a few human studies done, again, on like the population level, that shift workers, flight crews that are constantly undergoing jet lag, like they do have an increased risk for cancer and cardiovascular disease. So there is unfortunately lots of evidence out there to support in a chronic fashion. And of course, it's not true for everyone, right? Like some people could work graveyard shift for 30 years and just be fine. But coming back, Dr. Hopkins, to what you guys are working on at Illumina, I think that's really the key to kind of harnessing the power of our circadian rhythms, that we might be able to look at someone's clock gene sequence and say, you really shouldn't work, the graveyard shift, and that it might help us, kind of better plan our schedules to harness the power of our genome and also our circadian rhythms.
I imagine there's been a lot of transcriptional studies looking at which genes are involved and can you target those genes? And I'm thinking like a pharma guy here.
Oh yeah, we have some funding for a pilot study to look in mice that have an acute kidney injury. We've done some preliminary work that causes disruption of the circadian rhythms in the kidney, and we're going to be testing with a sort of the equivalent of a pill, but for the mice, and it's a compound that activates another one of the clock proteins, and it's been studied in a number of human studies looking at treatments for, I think they've tested it for diabetes and also some sleep phase disorders. And so that is definitely on the horizon. I think that we might have, like a small molecule that some people could really benefit from.
Yeah.
That is really interesting. As someone who I'm a former teacher, and I just thought about your presentation where it said, the best hours, the hours in the day when we are our best selves. And I was like, that's the complete opposite of what like the schedule most most schools operate. And I could definitely tell the difference between the students that I taught in the morning and then the students that I taught in the afternoon. And I tried, some teachers I know that I work with, we try to schedule tests on different days, depending on when they saw their class. Like students in the afternoon might perform better on a test than students, taking the test first thing in the morning. So all of these have like really interesting implications for even like secondary education in this country. So in the US, we're speaking from someone who's in the US. So yeah, that's all
absolutely.
Really interesting. Yeah,
there's a group in Seattle that successfully lobbied their school district to have later start times for the high schoolers, because that age group of teenagers, they just naturally tend to be more in that night owl phenotype. And so they saw pretty impressive improvements in test scores and, how they reported fatigue, and it did make a difference.
Like mental health, everything, like it's all connected. I think sleep is super important. I know sleep is super important. It's important for me. So yeah. I will turn over to our first question. Says, does not sleeping enough or sleeping at different times disrupt our circadian rhythm and increase our risk for kidney issues? So yes, that would be towards you, Dr. Gumz.
I think there's definitely some evidence for that in my lab and in my collaborators' lab. So we did a joint project with a group at Morehouse University in Atlanta, and they actually used a rat model, and they did a model of shift work where they shifted the light cycle by 6 hours in a chronic fashion. And it did result in differences. It changed the circadian pattern of urine production, and also hormone excretion was another variable that they measured. So that's on the basic research side. We definitely see evidence of that. And then there have been some studies done out of Korea, actually, showing that shift workers, particularly men, have an increased risk for chronic kidney disease.
Thank you. Thank you for answering that, Dr. Gumz. I asked what was like, what you enjoyed the most. What do you feel is the most challenging aspect of your job?
Now, if I can go first, I think keeping up with the incredible pace of science and how much is changing and how fast it's changing, it's really challenging. I feel like for every, just everything is moving at a fast pace, which is great. Challenging.
It is. It's the fun part, but it is also a challenge. For me, I would say it's the grant application process. So all of my research is supported by funding from the American Heart Association or the National Institutes of Health. And so the way that process works, they sometimes, if you're lucky, they fund 15% of applications that are submitted. And so that can be incredibly challenging.
We have another question. It says, for Dr. Gumz, how would I be able to apply to work at your lab? Are there any summer opportunities? Your work sounds really interesting.
Oh, that's a great question. Yeah, I would say, that's another favorite part of my job is working with students. I mean, I keep getting older, but they stay the same age. So I feel like it really keeps you young, and they're, so excited. And of course, everybody in the lab, we're so passionate about what we do. So it is a lot of fun. So speaking of grants, I'm working on one with a group of people at the University of South Florida to support, it's like a summer research grants. I'm working on one with a group of people at the University of South Florida to support, it's like a summer research program, so that we would have opportunities like that. But I always tell students to just send me an email. And if I can tell that they're really interested in circadian rhythms, we kind of take it from there. So we do have summer students, and we also have undergraduates. And I think that we are now allowed to have high school students again. For a while, they were not allowing us to do that. But I think it might be possible again.
Awesome. It does sound fun, really. I have a thing with rodents, so I don't know about that, but obviously you can't work around that. So they tell us a lot about, the human body without practicing or doing experiments on humans. So I get it. I totally get it. But, not everyone is as grossed out by rodents as myself. Right. So yeah, those sound like really great opportunities. Thank you so much, Dr. Gumz and Dr. Hopkins, for your presentation, for helping us celebrate DNA Day. Let me share my screen if anyone has any other questions. I know that Dr. Hopkins provided his contact information before, and we provided contact information, for Dr. Gumz lab in the chat. So if you want to explore more about the work that her lab does, definitely check that out.
I was just going to say, I should have followed Dr. Hopkins' example, because I do have Twitter. My handle is at @MLGumz. So that's a fun place to talk science too. And I think it helps me keep up on the literature as well.
I think I pasted that in the chat. So the Twitter handle so if you want to contact her on Twitter, her handle is in the chat. And then we do have one last question. It says, Dr. Hopkins, you mentioned an internship, but are high school students able to do it now, or is that something for the future?
I think the internship actually is geared more towards college level internships. Go to the website that I gave the link to. Go search Illumina internships and see what's going on. And depending on where you are, maybe you can get involved.
Thank you, Dr. Hopkins. So thank you once again to our panelists, Dr. Michelle Gumz and Dr. Christopher Hopkins. I really enjoyed hearing what you both do. And once again, thank you again for discussing DNA Day, helping us celebrate learning about all the wonderful things that the human genome is capable of. You almost have converted me, Dr. Hopkins and Dr. Gumz. And thank you all for attending. We want to also thank our sponsors and Illumina for helping us make this event happen. Feel free to look at our website for upcoming events in May and June. The link is in the chat. And we will notify you when this recording is available if you want to go back and rewatch, relearn. So yeah, thank you again, everybody, for attending, and for our panelists, Dr. Gumz and Dr. Hopkins. Have a great rest of the day, everybody.